Okay, so what we're going to talk about today, general idea. We finished the synaptic transmission, right? I think so. Yeah. So we're going to talk about a little bit about neurotransmitters. I'll explain what you need to know. I will be referring to some extent to the lecture notes. Don't worry, this is being recorded. So we'll be able, if you have some questions, we'll be able to refer to it. We will talk about graded potential and action potentials, some neuronal circuits, stuff like that, minor things. And I hope we're going to start talking about functional anatomy of growth. Okay. So without further ado, okay. Well, let's talk about neurotransmitters. So remember our conversation, how signal is transmitted from one neuron to another. Neurotransmitters happen to be synaptic cleft, and then they bind to the receptor on postsynaptic neuron, right? Remember that? The neurotransmitters can be broadly divided into two large groups: the small molecule neurotransmitters, neuropeptides. Okay. Now you have tables in the PowerPoint, whatever it is. You have tables. A couple of slides for small molecule neurotransmitters, and I believe it is one slide for neuropeptides. I want to be very clear what you need to know. In terms of neuropeptides, small molecule neuropeptides, I want to be very clear on what you need to know for all of those groups. For small molecule, I don't know if you have those PowerPoints with you, but I remember it goes second page. On the second page, there is glycine. I think it's a third row from the top. First page and second page up to glycine. For each of those neurotransmitters, you must know whether they are excitatory or inhibitory or both. That makes sense. Everything that goes after glycine is basically FYI. Am I clear? I'm going to break it down very clearly. So yes, I can ask you, which of the following neurotransmitters is purely or mostly inhibitory? And if in the slides it says mostly inhibitory, get rid of most inhibitory. Does that make sense? Or, sorry, neuropeptides. There are two rows. One row, endorphins and enkephalins. Those inhibitory neurotransmitters. Another row, tachykinins and substance P. Those are stimulatory neurotransmitters. These two rows for neuropeptides, period. I will not ask you anything more than that. I promise, it's all on record. You can get, I know that I will not. Okay. I promise there will be no questions about anything that I did not mention. Michael. Now, when you will be reading, hopefully, through those tables, you will find that there are some examples of sort of medical importance of neurotransmitters. Does that make sense? For instance, I break down in a pretty substantial level the various pharmacological interventions that can happen to acetylcholine. Okay. For instance, I bring, and that's kind of an interesting one. So remember, we talked about presynaptic neuron and postsynaptic neuron, right? And that there are receptors on the postsynaptic neuron and neurotransmitter acetylcholine binds. This is your signal transmission, right? And we mentioned that it is absolutely essential to get rid of neurotransmitter once signal is over, to avoid exhaustion of the postsynaptic neuron, presynaptic, just, because signal has to be terminated. Basically, when you said what you're supposed to say over the phone, you're supposed to hang up. Makes sense, right? And I mentioned that there are three mechanisms to get rid of neurotransmitter: diffusion, which is not really important, enzymatic degradation, and reuptake. So for acetylcholine, the main mechanism is enzymatic degradation. Okay? If you're curious, it's acetylcholine, the enzyme called acetylcholine esterase breaks it down to choline and another acetic acid. Will I ask you this reaction? No, it's not a biochemistry. Okay? But here's the deal. Here's the story. And that's a pretty fun story. So imagine that you somehow can inhibit acetylcholinesterase. Okay? What's going to happen to amount of acetylcholine in the synaptic? Folks, it's either a lot or a little. And the more we communicate here in class, the easier it's going to be for you during the exam, because that really improves the learning. So if you inhibit the breakdown of acetylcholine, what's going to happen to the amount of acetylcholine in the blood? Yeah, it's going to stay up, right? How does this elevated level of acetylcholine in the blood, how will it affect the signal? Will it sustain the signal, or will it bring the signal to a halt? Sustain, bingo. So signal will keep on going, right? Because essentially this stuff terminates the signal, and if you remove the terminator, signal keeps on going. So what we just described is the mechanism of action for poisons that are known as organophosphates. I don't know which specialties are you looking at. Anyone is looking at nursing, and if you, or PA. I have nothing against dental hygiene, but you're not going to see this patient. You're looking at nursing the PA, and or nurse practitioner, and you decide to work in mostly rural areas, you may well see the patients with organophosphate. Organophosphates are pesticides used in the farm, and they inhibit acetylcholinesterase. So signal mediated by acetylcholine keeps on going through synapses. That leads to a condition known as sludge. The syndrome described as salivation, lacrimation, urination, defecation, gastrointestinal cramps, and emesis. Each of these things is kind of unpleasant by itself, right? Imagine all that stuff happening at the same time. Pretty nice. These organophosphates actually are so good that Germans in the 30s created kind of updated version of these organophosphates, or the organophosphates on steroids, which you may know as chemical weapons such as VX. Same principle, okay? Now, here's the important question. Imagine, and I really want you to think and answer. Remember, it's not an exam. There are no stupid answers, okay? If you're wrong, it's okay. You learn. So my question, you have a patient who has organophosphate poisoning. Essentially, this patient has taken some chemical that inhibits acetylcholinesterase. So now you're facing a situation when ACH is in the synaptic cleft, It constantly binds to those receptors and constantly produces the signal. Describe to me a pharmacological intervention that can help that patient. You have all the information, I promise. I'm not asking about the name of the drug. I'm asking about the principle. So what your drug should do. Any idea? So again, keep going. Say it louder. Excellent. Usually, and when I say excellent, really good job. Because usually people say, how about we just get some so-called esters? You can't possibly give that much. But if you give a drug, so here's the receptor, right? Here's the acetylcholine. If you give a drug that binds to the receptor instead of acetylcholine and blocks it, essentially ACH receptor inhibitor, you're good. Does that make sense? This drug is atropine, and it is in the medical kit for virtually any modern soldier in case of gas attack. And it is actually a treatment that is a treatment for organophosphate poisoning. Does that make sense? So this drug here will be called receptor antagonist. Does that idea make sense? Receptor antagonist. Now, conversely, well, we'll give you an example. So acetylcholine is a really important neurotransmitter, not just in the CNS, but in your brain. Actually, you know what? Screw acetylcholine. Let's talk about endorphins. Okay? Another example. Endorphins are large neuropeptides that are produced in your brain when you are under stress. That can be exercise, that can be fight or flight reaction, like acute stress, when you are in basically fight or flight mode. The function of endorphins is to inhibit pain. Okay? They also make you hot. Runner's high, all that kind of exercise-related stuff. They indeed bind to their receptors and make you hot because endorphins are naturally produced opioids that are made in your brain. Okay? Now, can you be addicted to them? People who exercise regularly and then for some reason stop exercising, they kind of feel a bit dumb. They don't have a full withdrawal syndrome, but yeah, you have that great feeling, and now you suddenly don't. Now, I mentioned the word opioids. You probably have heard about stuff like heroin, morphine, or fentanyl. Well, those drugs are also opioids. They are either derived from natural sources, like poppy seeds. That's morphine and heroin is kind of modified, or synthesized artificially. That's fentanyl. So what they do, you have a receptor, and you have endorphin, and you have the same receptor, and you have fentanyl. Okay? The thing is, I don't remember the exact numbers, but say if endorphin stimulates that receptor at the level 1, fentanyl will stimulate that same receptor at level million. Does it make fentanyl a bad chemical? Not really. It's a wonderful painkiller. The only problem is that it not only, works as a painkiller, it also makes people, high. So, and it's such a powerful action that it's really hard to resist to try it again. That's the mechanism of addiction. Does that make sense? On top of that, there's another problem. You've all heard the story about the bullet and pride wolves, right? At some point, what happened? When wolves actually came, what happened? Nobody heard it, right? Here's what happens with receptors. When people start using opioid drugs, whether it's morphine or oxycodone or fentanyl, the stimulation of the receptors is so powerful that receptors is a protective mechanism. They develop resistance. Make sense? So the old dose doesn't work anymore. This is why people who are addicted to opioids have to increase the dose aggressively. And this is why the withdrawal syndrome for those people, if they try to quit, is such a terrible thing. Because suddenly they don't have this exogenous stimulant, whether it's oxycodone or heroin, right? Their receptors, they are resistant to such extent that endorphins that the brain produces are simply not enough to get any stimulation. Does that make sense? Okay. Now you ask, can it restore sensitivity? Yes, it takes time. That makes sense? You got it? So in this example, fentanyl or heroin or morphine or oxycodone, this will be receptor agonist. Am I clear? I'm driving this whole conversation to the point that I will tell you what you can expect in terms of the question. So another mechanism that I want to highlight is reuptake, right? Okay. This is when neurotransmitter is taken back into presynaptic loop. Does that make sense? So if you inhibit reuptake, neurotransmitter will stay in the presynaptic, in a synaptic cleft, and the signal will continue to go. Does that make sense? So reuptake inhibitors work really well. So, and different neurotransmitters have different termination mechanisms. For instance, for acetylcholine, it's enzymatic degradation. For neurotransmitters like dopamine or norepinephrine or serotonin, it's a reuptake. You good? So if my memory serves me well, and I'm not going to ask you this pharmacological examples, what's the mechanism of action for Prozac? If my memory serves me well, Prozac inhibits the reuptake of serotonin. Okay? So more serotonin stays in the cleft, and people, they become calm. Serotonin is a inhibitory. Cocaine and methamphetamines, they inhibit reuptake of norepinephrine and dopamine. So this is why people who use those drugs are kind of jittery and very sort of upbeat, because both norepinephrine and dopamine are kind of feeling good in the transmitter. Does that make sense? So what I want you to kind of focus on is being able to do. Like in this example, when Jake correctly suggested to use receptor inhibitor to treat this condition. What I will do in one of the questions, maybe more than one, not 50, one or two questions, I will describe you the clinical scenario. Such and such drug inhibits reuptake, stimulates reuptake, or inhibits degradation. That make sense? What do you need to use? And I'm going to be using words agonist and antagonist. Do you understand the meaning of those words? So you will need to choose which drug, and I'm not going to give you the names of them. So like receptor antagonist, receptor agonist, blah blah blah. What are you gonna do? Inhibitor of receptor, stimulant of receptor, stuff like that. You know, which drug will you choose to treat that particular clinical scenario? Does that make sense? In situations like this, drawing a scheme box. Just for my. Okay, we good? Trying to think of that. Yeah, I think I'm good. Questions? Okay. So we're going to move on and talk about really a huge chapter, and it's probably the most challenging concept in the transmission of the nervous signal. We're going to be talking about the rate potential and the action potential. So in order to kind of, clarify, so to say, some things before we start, just we're going to do a quick background, if you don't mind, because there are certain things I just want to make sure that nobody is caught off guard and nobody's like, what the hell is it? Okay? So we're going to be talking about the movement of ions across the cell membrane. Okay? I want to remind you a few things. First, cell membrane is hydrophilic on the outer and inner surfaces and hydrophobic in the center. Does that make sense? So only hydrophobic molecules can cross it directly. Hydrophilic molecules have to have some kind of a channel, okay? And in this particular situation, for this moment, when I say hydrophobic molecules, I mean ion, okay? Does that make sense? For instance, sodium will need a channel to get from, say, outside of the cell, inside the cell. So far, we good? Following me? Um, Makes sense, right? Next thing, types of transport. So transport across the membrane, it's a review, okay? Transport across the membrane can be passive, that does not require any energy, and active, and does. So passive will be simple diffusion. That's your hydrophobic, okay? Hydrophobic molecules, okay? These hydrophobic molecules will cross the membrane directly, okay? And facilitated diffusion. Which will include hydrophilic molecules, okay? All right, so we got this, okay? What do you need to remember about passive transport? Please, don't dismiss it. You can't see my facial expression. I'm digging. Understanding diffusion and active transport is essential for me. And AP1, AP2, we're going to be using the movement of ions across the membrane all the time in many different instances. So remember, diffusion is always movement from the high concentration to low concentration. If that makes sense. And it doesn't matter on which side of the membrane high or low is. That is a rule. Now, for active transport, what you need is a pump, right? You need ATP, and it's a movement from low concentration to high. Okay. Right? Good. Now, last bit of preparatory information is this. I expect you to know the concentrations of ions outside and inside of the cell. Now, I don't remember if I made this pledge before, but my general approach do not ask to memorize numbers. And if you want to know how it correlates with concentrations, I'll explain. All you need to know is on which side of the member the concentration of a particular ion is higher. Am I clear? You don't need to know the numbers, just the relationships. Understood? So, 4 ions, you can do it. Sodium, chloride, and calcium, their concentrations are higher outside of the cell. So basically, it's extracellular, right? And potassium concentration is higher inside of the cell. Does that make sense? So this is why I drew that sodium diffuses into the cell. So far, does that make sense to you? Now, when I say you should know that, I call this type of knowledge a 2 a.m. knowledge. You wake up at 2 a.m., you may not know who you are, where you are, but you should be able to tell someone. Sodium, calcium, chloride are higher outside, potassium is higher inside. Got it? Good? Yes. Yes. I will not ask you, it's a good question, I will not ask you about any other ion. And thanks for the question, because this is in general. My biggest challenge now is to remember to cover things either saying, look, anatomy is on you, like those neurotransmitters, memorize it, that's on you. I'm not gonna say this in the class, right? But I at least told you what you need to know. That makes sense. And to cover stuff that I can ask you. But if it's not in the notes, if it's not said in during the review, if it's not on a study guide, it is never going to go. I 100% guarantee. The last time I was caught with something that was on the exam but not on the study guide was like 2.5 years ago, and it was by a complete accident. I forgot to put few terms on the anatomy study guide. And guess what I did? Those were basically 3 points. If you got those structures right, 3 points. But I corrected myself, so don't expect 3 points. Okay? Everything that's on the exam will either discuss here, or if it's more memorization, you can get it from me. And I mentioned. Okay, we're good so far? So now we're going to introduce the concept of a resting membrane potential. Now, one of the really tempting things that some people and believe me, including me at some point, want to do with physiology is to find some kind of deep underlying law. Does that make sense? You took math before. So you took math before. You look at geometry, Euclidean geometry, there are 5 axioms as far as I remember. Based on these 5 axioms, you can build a really gorgeous building of this plane 2-dimensional or even 3-dimensional geometry. You know, it is all, you know, self-contained and great science, right? And all you need is 5 axioms. That's it. Physiology isn't like this. So like certain things we observe, like we see, we measure, and then we try to come up with explanation. Why things happen this way? Why do we have this parameter or that parameter? Does that make sense? Understand? So we just observe things that are in the nature. So, if you will take a cell, okay, that's just one member. So that's out, that's in. And you will stick an electrode into the cell, okay, and you measure the electrical potential. You can do it. Measure the electrical potential inside of the cell. It turns out to be negative 70 millivolts. Do you need to know the number? No, you don't. I'm just illustrating. It's negative 70 millivolts. So the question is, let's try to kind of play scientists and figure out why. We already know that sodium chloride, calcium are higher on the outside, potassium is higher inside. So let's do this. We're going to hypothetically imagine that we have a cell membrane which contains only potassium channels. Does that make sense? So this is the membrane. That's your potassium channel. Okay? This is outside. This is inside. Will potassium diffuse? Channels open. Will it diffuse? What is it? Come on, yes or no? It's 50-50. The concentration of potassium inside of the cell is higher than on the outside. Channels open. Will it diffuse? Yes, yes, it will. It will diffuse. Here's your potassium diffusing out. What is the charge on the potassium? Positive, right? So tell me, please, when you start taking positive charges from inside of the cell, which charges start to accumulate inside of the cell? If you take positive out, don't be shy, please. Huh? Bingo. We've got negative charges accumulating on the inside. And outside, obviously, is going to be positive, right? Now we have positively charged potassium here outside, and inside is negatively charged. And what do positive and negative charges, how do they kind of interact with each other? Opposite charges do what? That's right. Let me say, please, it makes it really better. So eventually, potassium will start flowing back into the cell because of the electrical attraction. Does that make sense? So it gets out of the cell due to the basically chemical diffusion, but then it starts to flow back into the cell due to the electrical attraction. And at some point, the outflow and inflow will balance each other. Does that make sense here? So overall flow of potassium will be zero. Net flow will be zero. Does that make sense or not? Usually compared to a grocery store, like you go there in the middle of the day, the number of people that walks in and the number of people that walks out is the same. You're gonna have the same number of people in the store. That makes sense? It's not the same people, just the number. The net flow is zero. Does that make sense? So net flow will be zero at certain distribution of positive and negative charges across the membrane. Am I clear? So theoretically predicted potential, and this is the potential because positive charge negative. Theoretically predicted potential for potassium at which net flow of ion is zero is called Nernst potential for that ion. And for potassium, it's going to be, I think, 90 millivolts. Okay? We always measure inside. It's a rule. Potential is measured inside. So negative millivolts. That makes sense. And how about we do the same for sodium? Now it's your turn to tell me what I should roll. So concentration of sodium is higher where? Outside. Nothing. Masks, I couldn't imagine how muffling they are. I really can barely hear. I don't know how well you can hear me. I'm pretty loud. So sodium will diffuse where? In the cell, right? And then electrical forces will start pulling it out. Does that make sense? Good. So eventually now, when positively charged sodium gets into the cell, it brings positive charges in, bringing negative charges outside. So you can say that Nernst potential for sodium would definitely be positive. Make sense? Because we measure it here. So for sodium, Nernst potential will be not sure, probably, whatever. Doesn't matter. Not for the no. No. I promise you, if you need to know a number, I'm really thinking. I think we're done with numbers. It was rule of nines. That was the only number question that I asked. I don't think I asked any question that deals with actual memorization of numbers till the end of a course. From I try to analyze, I don't think so. Okay, no numbers. Let's say this. If you need to know, I promise, I guarantee you, I'll tell you. But I don't think you need to memorize any numbers until you get to it. What I'm trying to bring up, remember we're playing scientists. So now we need to take these numbers and somehow come up with 70 million. Now, why we consider only these two ions, sodium and potassium? Turns out that the overwhelming majority of channels in the membrane are either sodium channels or potassium channels. Calcium and chloride, they do not participate in the formation of resting membrane potential. Does that make sense? So the first inclination, of course, is to say, how about this? Since only these two ions, sodium and potassium, are important for the formation of resting membrane potential, we're going to take these two numbers, put them together, and that's going to be our answer, right? Negative 30. Nope, doesn't work. Okay? So if you would put them together, you're going to get -30. But the actual measured membrane potential is more negative than that. What it means? It means that potassium plays bigger role. Potassium is more negative, you see? Potassium plays bigger role than sodium. Do I make sense? And in fact, if you will check how many potassium channels are in the membrane, there's simply more potassium channels than sodium. Am I clear? No. Awesome. So this is your resting membrane potential. It means that at rest without any stimulation, cell has a membrane potential of -70 millivolts, and ions that participate the most are sodium and potassium. Does that make sense? So we're going to be focusing on these two mostly. We're going to be talking about graded potential reaction. I have one question. It's not trivial. It's not hard. Okay. I just want to see if you can get this, because it shows like whether you got this whole thing right. Really, not a complicated question. You have all the information to answer. Imagine an absolutely hypothetical situation. We have a membrane that's out. And I'm not saying that this situation actually happens in life. I created completely hypothetical. The concentrations of, I don't know, sodium, whatever. Sodium outside. Inside, the concentrations of these ions outside and inside are equal. Make sense? Have a membrane, on both sides, concentrations of sodium are equal. Simple question. What's the Nernst potential? Yep, 0. Good job. There's no flow. Doesn't shift. That makes sense? That's it. You good? Any questions about this? Go ahead. It is, again, it, so it, I hope I understand your question, because and there's no insult here. So Nernst potential is a theoretical parameter. You can calculate it, and it depends on the difference of concentrations outside and inside. The reason why at some point net flow becomes 0 is because chemically driven diffusion is counterbalanced by electrically driven movement of ions. Did I answer your question? Thank you. Good? So now we are equipped by this knowledge, and we're going to talk about graded infection structures. So first, we're going to address the issue of a graded potential. Well, first we need to address the issue of different types of channels. Channels B. leaky. I think I don't need to explain what leaky means, right? So leaky channels, ions simply just go through. Does that make sense? Like the door opening in the wall without the door. You can just go through. You don't need to open it. Does that make sense? And then we have gated channels. We have ligand gated channels. We have voltage gated channel. And we have mechanical gated. Okay? I'm gonna start from here. So mechanically gated channel. What do you think opens this channel? It's mechanically gated. Mechanical stimulus. For instance, pressure, vibration, right? Does that make sense? These channels are used in our sensations of things like pressure, touch, vibration, that's distension. Good? Now, voltage gate. This I will explain. So remember we mentioned negative 70 millivolts, the resting membrane potential, right? So imagine that the resting membrane potential, the channel is closed. If membrane potential changes in a certain way, it will become open. Does that make sense? Finally, ligand gated channels. So here, we need to go back to our discussion of synaptic transmission, and we mentioned that there is a neurotransmitter, okay? And it binds to some kind of a receptor, okay? So essentially, ligand gated channels represent one type of such receptor. So receptors for neurotransmitter can be divided into ionotropic and metabotropic. So what is ionotropic receptor for neurotransmitter? That's your ligand-gated channel. Let me demonstrate. So this is the membrane, right? This is the channel that is coupled with a receptor. And this channel is closed right here. Okay? Make sense? So suddenly, neurotransmitter, not suddenly, but neurotransmitter binds to the receptor. Okay, that's blue stuff is neurotransmitter. What happens is, in response to that binding, channel becomes open. That's your ligand gate. Don't now look. A lot of things here are really simple. Don't overestimate the complexity. Think of it this way. This is the ligand. This is the receptor. I know that folks on the camera cannot see me, but what I'm trying to do, I'm trying to open the door with my key, electronic key. Okay, so this door is closed, and this is your receptor mode, right? And this door is ion channel. This is going to be ligand. How fast does it go? Pretty fast, right? Right away. So ionotropic receptors, usually they respond very quickly. That makes sense? Because the neurotransmitter binds, boom, they all. Ions start to flow, shit happens. Okay, we good? But the effects of ionotropic receptors, the effects on postsynaptic neuron are very short-lived. because, channel opens. The door is locked already. That's a great example. Okay? Opens, boom, it closes. Does that make sense to you? Now, what is a metabotropic receptor? In the membrane, you have a receptor. Okay? The neurotransmitter binds to the receptor, and this binding activates the cytoplasmic protein that is called a G protein. You see a little asterisk? Asterisk means that G protein is actually activated. Does that make sense so far? So this activated protein then moves to an enzyme. And if you're curious which one, there's more than one. This is why I call it enzyme. Okay, moves to the enzyme and activates that enzyme right here. And what is the role for that enzyme? This enzyme produces second messenger molecule. That makes sense. No? Second messenger, the easiest analogy is when you go to your friend's house and you ring the bell and your friend's sister opens the door. And you don't want to take off your shoes and you say, hey, can you please call Bill? Okay? So your sister, who doesn't have time to call Billy, because she's too busy, she calls youngest brother and say, hey, go get Bill. Okay? So that's you. That's kind of a sister. And that second messenger is Billy. No, not the Billy, the younger brother. Okay? So it takes a lot of steps, right? Signaling through the metabotropic receptors, neurotransmitters, is usually pretty slow. But the effects last much longer. Does that make sense? Do you follow? And I'm going to give you an example. Typical ionotropic receptor is a receptor for acetylcholine. And acetylcholine is pretty much an essential neurotransmitter for your skeletal muscle movement. Now, think of your skeletal muscle movement. Are they quick? You play sports? Yeah, they very sharp and quick. But the thing is, after, I don't know, you take a swing on a ball, if you play baseball, softball, or you kick the ball to play soccer. Okay, you do it. Your leg or your hands, they do not stay in this position. You send them the signal, they did whatever they're supposed to do, right? And they relax. They go back. It all happens very quickly. Does that make sense? That's your ionotropic effect. On the other hand, imagine that you get in some stressful situation, you have that fight or flight reaction, right? Your heart rate and blood pressure go up, you start sweating, time out, blah, 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 blah. Situation's over. You still shape. That's norepinephrine. It works through metabotropic receptor. Does that make sense? So the effects stay for a while. So far, we're good? So let's see. Oh, yeah, you ask, okay, second messenger, so what? Second messenger can do different things. The postsynaptic neuron, for instance, opening the channels. And just, how these channels on the postsynaptic neuron are open. Does that make sense? For our intents and purposes, we're going to be focusing on ligand-gated ionotropic receptors. Can I kill this stuff? Okay. This is your cell, neuron, okay? Dendron. Remember, negatively charged inside, positively charged outside. You with me so far? So here, we've got some kind of stimulus. Mechanically gated, with mechanical stimulus, chemical stimulus, doesn't really matter. Stimulus. This stimulus opens sodium channels. Sodium starts to flow inside, right? Bringing positive charges with it. And what we see that the patch of the membrane, hold on a second, didn't want to do that. So part of the membrane gets some, extra positive charges inside and extra negative charges on the outside. Does that make sense? So first of all, this feature when resting membrane potential, or membrane potential, should I use this? Membrane potential becomes more positive called depolarization. Okay, why? Because essentially, originally negative inside, that polarized membrane, as positive ions flow in, it becomes less and less polarized. It becomes closer and closer to zero. Does that make sense? Got it? So membrane becomes depolarized. It is really, really depolarized. Here, maybe, the side of the stimulus. And then, depolarization, that change in a charge distribution eventually goes away. So no depolarization on the side. There are leaky channels. Sodium goes back to the side. So it basically kind of fades away. Good analogy of that situation. You take a stone, throw it in the water, and there are waves. Waves are the highest at the place where stone hits the water. Does that make sense? And then they eventually fade away. Does that make sense? So, do you create some kind of a potential right here? Yes, you do. The stronger the stimulus, the more sodium channels are going to open, right? So the bigger the potential is going to be. Are you following me? So this is why it's called graded potential. You can represent it actually like this. So if this dashed line is resting membrane potential. Let's say you have, okay stimulus. That's your okay graded potential. Now then you have a stronger stimulus. Well, I agree. Eventually it will go. Does that make sense? I'm going to explain the concept, and I really want to kind of get it to you. But graded potentials can have different values. The higher the stimulus, the higher the graded potential. That makes sense. Now let's take a look back at the membrane and see what happens in one really interesting exquisite case. This is your ligand-gated channel. Okay, sodium channel. This is your neurotransmitter right here. And it opens that ligand-gated sodium channel. So it is right here. The sodium starts to flow into the cell. Make sense? So the inside becomes locally depolarized. Depolarization. Good? So the charge on the membrane, it changes, right? And since charge changes, this change in voltage can open, and I'm going to prove that it's ligand-gated channel, right? The change in charge can open. So this is going to be voltage-gated channel. Okay? And the sodium, so far we're talking about sodium, starts to float into the cell. Does that make sense? Are you following? So it causes local depolarization. So that next voltage-gated channel opens and sodium starts to flow into the cell. Does that make sense? So if, and it's important, if depolarization here caused by initial stimulus is big enough to open voltage-gated channel, then depolarization here will open next voltage-gated channel, and then next voltage-gated channel, and then next voltage-gated channel. Does that make sense? I see some confusion. Go ahead. See? All what opens them? Change in the membrane charge. See, your membrane here, without any open, it's negative on the inside, positive on the outside. You give it some stimulus. You follow me? Give it some stimulus. That stimulus causes the influx of positive charges into the cell. So the membrane potential goes from, let's say, negative 70 to negative 30, makes it more positive. Okay? When it hits that negative 30, that's enough to start opening voltage-gated channels. They are all signal for them is the change in voltage. Does that make sense? Change in the charge levels. Did I answer your question? Sure? Good. And when this channel opens, it changes the charge further. If it's enough, next channel opens and next channel opens. Does it remind you of something? So yeah, really fascinating. Sometimes they do massive displays of those games and being done with. And I use analogy to help you to understand better how it works. Imagine you have a set of diamonds, all placed properly, on the side, so they can one flip another. You know what I'm talking about? Those are not your average diamonds. Those are massive, 10 feet tall, 1,000 pounds each diamond. Okay? So you come to that domino and you push it. Nothing else. 1,050 pounds. Right? And I think, okay, and you push it harder. It starts to shake. Is it going to fall? Not now. That's your shake. You push it even harder, shakes more, doesn't fall, eventually stands still. That's the green, the bigger gradient potential. And then you just run into it and slam it. And it finally falls. And when this domino falls, all others are going to fall. Does that make sense? So what I'm trying to tell you, is when the signal is going to reach a certain threshold. It's going to become an action potential. So it's going to hit all the way through the top. Your domino is going to fall. Does that make sense? Signal will move through. Does that make sense? Now, what would be a good physiological example? Can you imagine, hypothetically? Well, probably not so. Okay, I use this example in my life. Imagine a speckle of dust landing on your skin. How are you going to feel it? One speckle of dust. Most likely not. Will it stimulate some sensory cells in this? Probably so. It will create this greater potential. It's not going to go anywhere. Now, instead of a tiny little speckle of dust, you have something, also dust, a bigger particle. Okay? So stimulus is stronger. If the response that is produced by tactile receptors in your skin hits the threshold, you feel it. So what I'm trying to say, there's a bunch of stuff that constantly lands on your skin. You don't feel it. Because that stuff does not produce necessary level of stimulus. Does that make sense? Now, few features about action potential before we move on to why it has such a shape and whatnot. Action potential in the neuron, if we draw a neuron like that, so that's axon, there's a dendrite. Action potential is formed most and foremost right here, in that zone here. And it travels down the axon. Very clear. So graded potentials will be formed on the synapses, on the body, but eventually the action potential will be ultimately formed on the zone. Is that clear? Those dominoes that I described, can they fall half-ass? Can like do this and nah, not going anywhere. No, right? So dominoes either fall or not. Am I clear? Action potential is all or none event. Makes sense, right? And the fall of a domino, it cannot fall farther. It was vertical, now it's horizontal. Does that make sense? So action potential has a fixed value. It's always a certain voltage. That's all or none principle. Does that make sense? So whether it was a tiny, I don't know, whether it was a drop of water on your skin or a whole shower, or I don't know, okay, whether it was this or a slap on the wrist, surprisingly, the voltage that goes through your nerves is going to be the same in all cases. You can ask, I'm going to say, I can feel the difference between the intensity of this versus this, right? Well, think of the dominoes. They all will fall, but what can change if you slam it hard and then if you slam it really hard? That's kind of more complex question. You kind of have a wild, you have to have a wild mind. They can fall like this, right? Or they can fall much faster. Make sense? They're still going to fall, but it's going to be kind of different rate of each fall. This is how the strength of intensity of a stimulus is modulated. The stronger the stimulus, the more frequent action potentials are. Does that make sense? So I use this comparison if I speak in more quiet voice, action potentials are going to go like this. And if I raise my voice to louder sounds, frequency of action potential is going to go. Does that make sense? And just basically, you can think of it this way. How we distinguish between the signal and no signal. Well, in some cases, action potential and no action potential. And in some cases, you have the baseline frequency. Make sense? And if it becomes more frequent or less frequent, that's the signal. That's going to be really important when we're going to talk about special senses, hearing, balance, stuff like that. Okay? Let's take a look. Because the stuff that I'm going to talk next is like 10, 15 minutes. Let's take a look. Okay, here we go. We're going to try to imagine today. Imagine that you have a neuron, okay? An axon, to be specific. And you take an electrode and you measure changes in the membrane potential in a specific patch of the membrane. Make sense? Not one point, but like a patch. There are going to be few, channels, sodium channels, potassium channels here and there. That make sense? By the way, this method is one of the most common neurophysiology methods that is called patch clamp. Yeah. We can reflect changes in membrane potential on the graph. It's going to be time on the x-axis, and it's going to be voltage on the y-axis. This is going to be resting membrane potential. And this is going to be the threshold. That makes sense? So we're going to start with opening of sodium channels. I deliberately, most likely, these channels are going to be voltage gated. So here you have voltage gated channels opening, right? As they open, sodium flows into the cell. Voltage becomes more and more positive. It hits the threshold. You get yourself a full-blown action potential. Okay? So if you remember our previous conversation, this depolarization, let's call it depolarization. D is called depolarization. Cell becomes more positive during depolarization. This depolarization happens due to which ion? More positive inside of the cell. Becomes more and more positive inside of the cell. Which ion flows into the cell? Take a wild guess. Huh? No. Positive ion flows into the cell, right? Because the membrane potential becomes more positive. But which ion will it be? Which positive ion? Sodium. So we can say sodium in. That makes sense. Now, all channels here are going to be voltage gated, so you don't have to worry about it. Now, what happens here at the top? Those voltage gated channels, interestingly enough, close. So sodium channels close, while potassium channels open. Right? And potassium starts to flow out of the cell because concentration of potassium inside the cell is higher, blah, blah. Got it? So that's going to be potassium out. We call this process repolarization. Okay, so we can write down the potassium channels. Oh. Now the deal is, look at this. So membrane potential right here, can you see that? It reaches resting membrane potential. Got it? See that? Almost there. Almost there. What would you expect to happen? That it kind of, flatlines again, properly, right? Fortunately, it doesn't. Or actually, not unfortunately. Fortunately, it doesn't. Potassium channels are slow. So although cell hits the resting membrane potential, they remain to be open. Are you following me? So membrane potential continues to be negative. Okay? Make sense? This part, actually, let me use slightly different color. This part right here is called hyperpolarization. And cell becomes more negative than the resting membrane potential. Does that make sense? So hyperpolarization happens because potassium still flows out. Now at this point, right here, finally, potassium channels close. Does that make sense? Now, mind you, this whole situation in the cell is a freaking mess. Right? Are you following? Why? Because sodium left the cell. The sodium has flown into the cell. Potassium has flown out of the cell. Concentrations of ions are in disarray. We need to restore them. The restoration is the responsibility here, brown part. It's a sodium potassium pump, which restores resting membrane potential. Does that make sense? You following me so far? So this is the typical description of what happens at this patch of the axonal membrane when action potential sweeps itself from cellular body to the synaptic terminus. One more time. When it arrives on that patch, voltage gated, sodium channels open, sodium starts to flow into the cell. Once the membrane potential, the action potential reaches its peak, sodium channels close, potassium channels open, potassium starts to flow out. This is called repolarization. Repolarization goes on, and eventually, since potassium channels are slow, cell actually hyperpolarizes, okay? And eventually potassium channels close, and sodium potassium pump restores the proper concentrations of ions and resting membrane potential. Does that make sense? It's kind of a long story, but everything checks out. Now, remember I told you we're going to be playing scientists, just, asking yourselves different interesting questions. So one interesting question. Remember we said that, the signal goes from body to the terminal. Does that make sense? Why? Like, why can it go backwards? Why can't it just, go this way, then this way? What prevents it from doing so? Here's the thing. Turns out that the sodium channels close. They aren't just closed. They are blocked. two analogies that I use to kind of describe. One is this. Imagine that you are running down the hallway, and there are plenty of doors. Make sense? In the hallway. So technically, you can go there, you can go back. You understand that? Now imagine that once you go through the doors, they close behind you and they stay locked for 5 minutes. You can't really go back. The only way you have is forward. Does that make sense? Do you understand what I'm proposing? Another example that I usually use, I kind of faced it. With a parking lot next to my workplace. And I swipe the card and I park myself, and I wanted to park my wife's car for 15 minutes. And I go back, like I go through the gates, I go back, I swipe my card, nothing happens. This gate, after you swipe your card, is locked for 5 minutes, so you're not gonna have like 1,000 people on the parking lot. Does that make sense? So this is the idea behind locking the sodium channel. Once they close for a certain amount of time, they cannot be opened by anything. Does that make sense? Which means there is a period of this, which is called absolute refractory period. Absolute refractory period, by definition, is the period in synaptic transmission when no kind of stimulus can cause action potential. Does that make sense? Remember my example with flapping? Remember that? I'm really into heavy metal. So, does anyone? Well, I know. The drummer of my favorite band holds the world record for the fastest hand drum. No. You'll never guess. 1,000 and a change beats per minute. Hands. There's a video. Okay? Now, I think it's kind of a limit of human possibility, right? What I'm trying to say is that you're going to do like 1,200 bits per minute. Fine. But there's always going to be a little pause between the hits. Does that make sense? So this absolute refractory period makes sure that first, there is a pause between the action potentials. Second, that action potential does not go back because, once the sodium channels close, this whole wave can only go to the right. That makes sense? Now, what about this part? At this point here, sodium channels can be open. They're not blocked. They can be open. Look at this. How far do you have to go here to reach the threshold? This far, right? See that difference? How far do you have to go here? Much farther. Does that make sense? You see the difference in those distances? So this part right here is called relative refractory period. Due to this hyperpolarization caused by potassium efflux out of the cell, action potential here can be generated, but it requires a stronger stimulus. Does that make sense? In order to hit that threshold. You got it? Makes sense, right? So think of it this way. This picture represents a certain frequency of action potential. And remember, frequency relates to the strength of the stimulus. if stimulus becomes stronger, then action potential, next action potential will be generated not here, but here. Frequency will increase. Stronger stimulus, frequency will increase more and more and more. Does that make sense to you? Got it? Okay. Now, you may wonder, okay, I'm going to ask a lot of questions from this. So, few advices. One, not only be able to draw it, but understand what's going on. Because questions will be, which of the following happens during depolarization or repolarization? From my experience, just memorizing doesn't work. You need to understand what's going on. Second advice, when the exam comes, get the paper. I can do the stretch paper, draw it. I allow it, it's totally fine. As long as you're not using any props, right? You know what I'm saying? Like I give you Scantron, exam, you have your pencil, you're more than welcome to start drawing that kind of stuff. And then you use it to answer questions. It's totally fine with me. Okay? Are we good? Now, a few things about the action potentials, transmission, and everything. So first of all, remember that picture of the neuron that we created. So these are dendrites, right? And here's going to be graded potential. Can this graded potential create an action? Hypothetically, yes. In most cases, no. You need several graded potentials that will be added together on the axonal helo. This is where action potential is going to be formed. Are we clear? Good. Second, how do we call this action potential? This graded potentials. So if graded potential depolarizes, makes it more positive. It's called excitatory postsynaptic potential. If graded potential hyperpolarizes, it's called inhibitory postsynaptic potential. Now, already figured that influx of sodium can depolarize the membrane, right? So influx of sodium can create excitatory postsynaptic potential. Am I clear? Now, it's not a hard question. It just requires maybe going back a couple of pages and looking at which two ions can create inhibitory postsynaptic. Which two ions can make the cell more linear? Again, you're nailing it. Potassium and chloride. If potassium flows out, it's positively charged. Positive charges are leaving the cell, so it becomes more negative. Chloride, it flows in, making cell more negative as well. Make sense? Good? Now, what if you have one graded potential, excitatory, and another inhibitory? What do you think is going to happen? One depolarizes, another hyperpolarizes. They're going to do what? They're going to cancel each other out. And finally, we're going to talk, not finally, we're going to say a few words about so-called summation. Okay? So summation can be temporal and special. Okay. So what is temporal summation? I'm not a better artist than the ones that illustrate textbooks. I'm not even going to attempt to do that. Here's my knowledge. So imagine you have one synapse, okay, here. You see that one synapse. And you get graded potential that comes in, goes away. Now imagine the frequency of these graded potentials increases and they start to overlap. Does that make sense? Eventually they overlap so much that they climb all the way, their sum climbs all the way to the threshold and triggers action potential. Anyone ever had a car stuck in the snow? So if you just approach your car, do this, and walk away, it's not hell, right? There are basically two ways. One way is to push it, and like you give it a push, and as it moves forward, give it another push. So you do really, really frequent pushes to your car. Does that make sense? Hoping that your efforts overlap, producing an impulse that will kick your car out of the snow. Make sense? This is temporal summation. Multiple stimuli over time on one synapse. Now, spatial summation is when you have multiple stimuli at the same time on different synapses. That would be a situation when your friends come to help you with a car and you all push it, you all push in different places, but you all push it together at the same time. Does that make sense? Know the summation. Okay? Three things that I wanted to chat about in kind of last bits. Saltatory conduction, neuronal circuits, and God. Facilitated and discharged. So what is saltatory conduction? Ages ago, last week, we were talking about myelination. Remember that? Maybe even before that. Myelination, rings the bell. Myelin sheath, electrically insulated. So now look what happens in non-myelinated neuron. If you have non-myelinated neuron, in order for action potential to move, and that kind of harkens back to your question that you had about like those sodium channels. So I have a sodium channel here. So I have a bunch of sodium channels. So you open this one, depolarization, right? Then depolarization opens the next one, next one, next one, next one. So this is an example of our dominoes that stand next to each other. You know, in non-myelinated neuron, you have to position those sodium channels really close because depolarization leaks away. Does that make sense? When membrane becomes polarized, those ions will leak back. It's totally fine because they're liquid channels and stuff. So you have to position those sodium channels really, really close to each other. Does that make sense? So in order to get from here to here, for action potential, you will need to open 5 sodium channels. Are you with me? Now imagine I will have to C2. That's non-myelinated. Now imagine you have a myelinated neuron. So that's your myelin sheath. Pretty much same distance, right? You have sodium channel here. You have sodium channel here. You have another one. Do you see that? Why do you not need any sodium channels here? Because myelin sheath prevents ions from leaking back. Does that make sense? Once you have depolarization, this depolarization is maintained all the way to the next sodium channel. Does it make sense to you? Imagine that by some magical effect, the domino falls on the ground and it's such an enormous earthquake that it drops next domino in like 50 meters. That makes sense? So here, to get action potential over the same distance, you need to open them in two channels. Does that make sense to you? Got it? So in this case, action potential jumps from one node of Ranvier to another. It's called saltatory conduction. So in myelinated neurons, conduction is saltatory from node to node. Does that make sense? In non-myelinated, it's basically continuous transaction. Question: which of these processes will be faster? This or this? Bingo, because all you need to do is to open only two channels, right? Instead of 5. Does that mean that myelinated neurons are better? No, they're just different. If you need to pass signal fast, like acute pain, okay, or skeletal muscles, myelinated. If signal may travel longer, non-myelinated will do it. Does that make sense to you? Like regulation of your digestive activity. Who cares? Your stomach will start churning like 15 seconds, not right away. It's fun. Does that make sense? Neuronal circuits. You have a wonderful illustration there. I'm going to kind of explain what those mean and functional role. So there are divergent circuits. So I'm one neuron, right? So you're going to be two neurons. Right? You are going to get the signal to those 3 neurons. Those 3 neurons will get the signal to 7 neurons. The signal gets spread. Does that make sense? That's a divergent circuit. Heavily involved in motor activity. Skeletal muscles. Okay? Convergent circuit. Those 3 neurons concentrate signal on these 2 neurons, and these 2 neurons transfer the signal to. Signal converges on one neuron, mostly sensory stuff, to amplify the signal. Does that make sense? Reverberating circuit. Katie, LaPortia, me. Signal goes like this. Often repetitive activities, like walking. And finally, parallel after discharge. Signal goes through me, goes through LaPortia inequality J, okay, and through Katie and Dylan to Jake as well. 2 processes go in parallel. Does that make sense? Higher mental activity. Calculation. Are we clear? Now, you should be able to match a description of a circuit with its name. Am I clear? Question can be, what is the correct description of reverberating circuit? Or, description is blah, 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 blah. What kind of circle do I describe? Does that make sense? Discharge zone and facilitated zone. I'm a neuron. I pass the signal to 5 neurons that are in front of me. Okay? Now, while I may bring these 3, so LaPortia, Jake, and Katie, I bring you to the action potential so the signal keeps going for you, okay? I failed to do the same with Dylan and Nicole. Does that make sense? So, but I bring those two closer to the threshold. J, Kate, and LaPortia will be in the discharge zone. Signal keeps going. Nicole and Dylan will be in facilitated zone. I brought them closer to the formation of action potential, but not quite there yet. So they need some extra cue. Does that make sense? Got it? Any questions? Okay. So you see these models? First of all, your exams are going to be based. Oh, Jay, there are some models there right behind Nicola in the back row that are not so fully disassembled. Okay, disassembled, let me check. So what I want to do is to review brain anatomy using these models and provide you some kind of a structural outline of sorts, structural functional. It's okay, they're plastic. They're really hard to break. That happens all the time. Don't worry. Your brain parts floating around. You just close to the entire cerebellum. It's okay. So we're going to try and since we're talking about central nervous system, let's talk about the parts of central nervous system. And please understand, some of the stuff that I tell you here is like action potential. I mean, that's a concept that everybody agrees upon. Nobody tries to contest it, blah, 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 blah. The breakdown that I give you, kind of sort of my copyright. I think it's easier to understand the structure of the brain this way. Does that make sense? You may see it differently in a different book. Totally fine. So we're going to start with CNS. We're going to start with the brain. Okay? Then there's a brain stem. And then there is a spinal cord. We're going to be adding some more here, so leave some space. So brain, that's your conscious processing, okay? From your personality to conscious appreciation of sound to recognition of images to a language, I mean, you name it. Right, clear? Brain further can be broken down into, another name for brain is cerebellum. Can be broken down into cortex, white matter, and basal nucleus. If you look at these models, you see this creamy surface. That's cortex. Okay. Deep in there, you don't really see it because it's deep in there. Under cortex is white matter. And the cortex, I'm going to put here conscious activity. Okay. Okay. White matter. White matter is due to the protein myelin. You just mentioned, what is the role of myelin sheath that surrounds neurons? What does it do? Electrical insulation. And which part of the neuron requires electrical insulation? Only. axon, transmission part. So white matter basically consists of myelinated axons, and it is responsible for signal transmission. And then you've got basal nuclei, which are really cool structures. Movement control. Are you with me so far? Now, let me ask you this. You're writing, right? Or you're walking, or playing some sports. Do you think you are consciously in control? I don't want to curb your enthusiasm, but not really, not that much. What I'm trying to say is that there are a couple of structures that provide the highest level of motor control, and you have no idea what they like that they want. The basal nuclei, and I'm going to put it here, cerebellum, or parental brain. So this is your cerebellum. You see the brown stuff here? Basal nuclei are basically deep there, right about here, deep in the brain, right here. You can see them on the model. So what they do, cerebellum makes your movements precise. So the mere fact that I can tap on that image and not on any other image, the mere fact that I can grab this model and catch it, that's cerebellum. That make sense? Now, basal nuclei prevents movements that are unnecessary. Without it, I would be going like this, okay? I would be just making movements that I don't want to. Does that make sense? In a disease called Huntington's disease, that's exactly what people develop. It's a condition known as chorea, and they have uncontrolled movement. Does that make sense? Now, there is a structure which I tend not to include in the brain. It's called Diencephalon, which in turn consists of three parts. Epithalamus, I'm not going to assign any function to it. I mean, it does in you and me at our age, and yes, I'm like 2 times older than everybody is, right? But in you and me, this structure is not really functional. In kids, it helps to maintain sleep-wake cycles. In adults, not so much. Thalamus, which is a relay for information. Thalamus essentially decides where particular information should go in which part of the brain. You following me? And the hypothalamus. You can see diencephalon in the models right here. So this little thing that I'm touching, that's hypothalamus. That part in the center is thalamus. And epithalamus is, it's really tiny where my nail is, like, seriously, I'm not gonna, I'm gonna say a few words probably. I mean, it's really kind of, it's active in kids, in adults not so much. Good? Hypothalamus. Visceral controls. Hypothalamus essentially controls all physiological, all autonomic functions in your organs, from directly or indirectly, from heart rate to production of hormones to responses to emotions like, you know, elevated heart rate, sexual drive, stuff like that. Sexual responses, for instance, when somebody is sexually aroused, so on and so forth. Make sense? It's a primary visceral organ. Okay. Now I said directly or indirectly. Turns out that the thalamus can work through brainstem. And brainstem contains three parts. I'm going to go from top to bottom. The top part right here, it's called midbrain. Midbrain controls visual reflexes, startle reflexes, and startle reflexes. I'm going to do this and you're all going to, yeah. Visual reflex is when you follow the object. The best demonstration for visual reflex are bleachers at American Idol, USO, or basketball. People moving from side to side. That makes sense? Good. Right. And some pain control. Next one is Pons. That bulb right here. Respiratory controls. And finally, the lowest part next to spinal cord, medulla oblongata. So here you have medulla. Controls a bunch of visceral reflexes. When you're swallowing, salivating, hiccuping, coughing, sneezing, vomiting, urinating, pooping, digesting, it's all medulla. The heart rate is controlled through the medulla. The normal, like regular respiration, medulla. Does that make sense? And finally, spinal cord. Signal transmission, from here to here and vice versa, right? And somatic reflex. You familiar with the knee-jerk reflex? That's fine. Okay. That functional outline, that makes sense. Now I'm gonna be saying a few words about what I expect to study in the law. I think it's first 1 or 2 pages of lecture notes. And there you can find primary brain vesicles, secondary brain vesicles. So you should know, for instance, there's a secondary brain vesicle that is called mesencephalon. What does it develop into? It develops into midbrain. Telencephalon. What does it develop into? It develops into the cerebellum. Does that make sense? So there are a couple of tables. Yes, that's memorization. I'm not going to add any important information. It's all there. It's all in the lectures. It is recorded. I go over it in details. That make sense? Good? Any questions? Okay, so let's talk about brain anatomy relatively quickly. First of all, when brain grows, it grows inside of the cranium, and this is why it is folded. Do you see the folds on the surface of the brain? It is folded because it cannot properly fit into cranium, and this is why it's kind of wrapping around the diencephalon and brain stem. Now that folding increases the surface area, and increased surface area means more neurons in the cortex. You see what I'm saying? Cortex is on the surface. More surface, more cortex. More cortex, more neurons. Essentially, the more it's folded, the smarter you are. That makes sense? It was shown that in kids who grew up without intensive social interaction, there are less folds. Each fold like this is called a gyrus. And gyri, that's plural, are separated by these crevices that are called sulci. Now, there are some like central sulcus and lateral sulcus. You don't need to know it. I will not ask this on the announcements. Which sulcus it is, which chapter? Good? So brain is divided into two hemispheres, right and left. Hemispheres are separated by the median longitudinal fissure, or as one of my students put it, about correctly, right? Cerebellum and cerebrum are separated by transverse fissure. Okay, and each hemisphere has 5 lobes. 4 of them we can show. Frontal lobe, parietal lobe, temporal lobe, and occipital lobe. Lobes have anatomical boundaries, which, again, not the part of the exam. I find more important for you to know which function each lobe has. I'm going to talk about it, not today. For instance, like your visual cortices occupy most of your occipital lobe, so that's your vision. Or your somatosensory cortices, that's sensations from the skin and the muscles and joints, parietal lobe. Am I clear? So that's more what I'm going to focus on. Right. Okay. Brain anatomy, spine. So now we need to address some sort of accessory structures. And we're going to go back to function for a couple of minutes. Brain is very squishy. Basically, it's a lot of fun. It's very soft. Okay? Makes sense. So if you put a soft structure in a bony cranium, not a great idea. You shake it, all gets damaged, right? So you need to protect it. Protection comes from 2 different sources. One source are meninges. So what are they? Imagine that this here is the bone. That's your cranium. That makes sense? So first layer of meninges is called dura mater. So that's dura mater. Matter is, well, matter, and dura means hard. That's a dense regular connective tissue, really, really hard, okay? The function of dura mater, to kind of stretch out your brain, partition it, and affix it into the cranium. Does that make sense? Sort of imagine if I would be brain, dura mater would be rows that kind of hold me inside of the cranium. Okay, cool? Now here you can see 2 layers of dura mater. The one that clings to the bone is called periosteal, and the one that kind of moves towards the brain is called meningeal. Do you see that separation? That separation is called venous sinus. Venous sinus. Sinus is fluid. It's basically a blood vessel. So, sorry. Separation of the periosteal and meningeal layer creates venous sinus. Under dura mater is arachnoid matter. It's called arachnoid because it has this kind of fibers that look like spider web. Arachnes or arachne. It's a spider. Okay? And at the very bottom is pia matter, really, really thin and delicate. Pia matter immediately attaches to the brain. Now, there are a couple of spaces here. We have subdural space. Quite frankly, here, not much of a space. That's overly exaggerated. And we have subarachnoid space. Okay? So why do we care about specifically subarachnoid space? Blood vessels that feed the brain are in the subarachnoid space. You following me so far? There's a space here, there are blood vessels. If one of these blood vessels gets busted, blood starts to spill into subarachnoid space, which leads to subarachnoid hematoma, subarachnoid bleeding. Okay? Sometimes blood can spill. Sometimes blood can spill here. Got it? If it spills here, it's subdural bleeding, hematoma. Subdural bleeding. Why do we care? When blood starts to accumulate in any part of the meninges, leads to inflammation and increased pressure in the blood, which immediately impairs brain function. And depending on the location of subarachnoid or subdural hematoma, person may die really quickly. Best example, I believe it was subarachnoid bleeding, is Liam Neeson's deceased wife, who hit her head when she was skiing, said that I'm fine. Hours later, she was dead because of meningeal bleeding. Does that make sense? We're clear about it. Another important feature of subarachnoid space is that this is where cerebrospinal fluid is. Now, if you would look at your models, you may notice right about here, it's kind of a bit of a cavity. It's called the ventricle, a lateral ventricle. Then you have a bit of a cavity here. It's the third ventricle. And a bit of a cavity right in front of the cerebellum. That's the fourth ventricle. Now, we'll get to the ventricles in a second. Ventricles are cavities. Are you following? They are basically empty spaces. Now, imagine for a second that you take some plastic, gray plastic, you liquefy it, and you pump it into those empty spaces and let it become solid. Make sense? Then you clean up the brain matter, and this is what you're going to have. You're going to have a cast of two lateral ventricles, left and right. Third ventricle here in the center, between the hemispheres. Lateral ventricles will be connected to third ventricle by interventricular foramina. And here we see a fourth ventricle with the cerebral aqueduct. Now, if you would look, I don't know if you can see, may or may not see, there's some pink stuff. Okay, so this pink stuff is arachnoid, not arachnoid, sorry, choroid plexus. Sometimes I have a brain freeze and I just cannot remember the term. Choroid plexus. So in the choroid plexuses, you have ependymal cells. You may remember those. So those are ependymal cells. That's blood. So what these ependymal cells do, we get into the story about the fluid surrounding it, okay? These ependymal cells, they filter the blood, basically letting water and some solutes through, thus forming cerebrospinal fluid. Does it make sense to you? You got it? So now you have fluid that is constantly formed in the ventricles. This fluid will circulate around the brain and around the spinal cord, most importantly, around the brain. And it will essentially float the brain. You got the idea? So brain in your skull right now, it is floating. The layer of CSF that surrounds your brain is actually very thin. It's about 1 millimeter. If you want to see how thick it is, take a penny and look from the side. It's not big. Make sense? So your brain is constantly floated. It's cushioned in that CSF. So when you're moving around and jumping, your meninges and cerebrospinal fluid together prevent your brain from moving with the skull. When it does move, it hits the skull. Yeah, you get a concussion. That's it. We didn't talk about it. So far, we good? No, that's big. If fluid is produced, it's supposed to drain. Makes sense, right? So what comes from blood shall return to blood. This arachnoid matter forms the structure that is called arachnoid villus. See that? This is where CSF drains back into the blood. So cerebrospinal fluid is produced in the choroid plexus, circulates, carrying oxygen and nutrients to meninges and everything, okay? Circulates, and then drains into the venous centers. And that's a constant process. It's constantly produced, constantly drained. It produces about half a liter of fluid. Okay? Total volume is really small, about 150. Imagine somebody has a brain tumor, and this brain tumor, very unfortunately, squeezes the arachnoid villus. And the drainage of cerebrospinal fluid is okay. What's going to happen? You have a bathtub. You open the water and you put a stopper. What happens to the bathtub? Huh? Well, if you don't turn off the water, it's going to overflow. You know, that's a good thing about bathtub. It doesn't have a cover. Your skull, has this whole system where CSF circulates. It doesn't live in capacity. It cannot overflow. The fluid will start accumulating, mostly between the skull and the brain, right here in the meninges. Does that make sense? Increasing pressure in the brain. If you're a baby, newborn baby, it's a problem, but not too big of a problem, because your cranium is soft, and it will be able to expand under that pressure. That makes sense? If you're an adult, you bone. I mean, not completely, but it's pretty bad, because the bone is not going to give in. Your brain will have to, and it doesn't sound right. So how do you treat the condition that I just described? It's called hydrocephalus. How do you treat it? If you have one drainage block, what do you need to do? Like ASAP, in order to relieve the pressure. Yeah, you need to drain it, right? How do you do it? The easiest way. Drill a hole. No? Yeah, you can drain it like that. The only problem is, you can't leave the hole in the skull open. It's kind of a bit dangerous. And you can't put a hose it's clear to dangerous. So actually, surgeons will install if the problem with, let's say it's a brain tumor. The brain tumor will be removed, hopefully the patient will be fine, no more blockage, everything's all right. Does that make sense? But in some cases, what surgeons will do, they will install a drainage that will drain through the spinal cord. And I know, does anyone know where it is regularly drained? You'd be surprised. So there's a drainage that goes all the way from CSF through the abdomen. Make sense? Because then it can easily kind of disappear. It's pretty simple. Make sense? Question? Good. Next week we're going to chat about different cortices in the brain, and we can do different parts. I don't think that our exam will be even. I think 2 weeks from now sounds too optimistic. How about 3 weeks? Make sense? Good. Awesome.